Okay. So that I don't actually know who's all out there, and if your microphones are muted now, that's fine. But if you want to interrupt me, I realize it can be kind of hard in these kind of Skype situations. I can't like see your body language to tell me that you need to say something or you have a question. So please feel free to interrupt at any time. It's really not an issue. That I think I'm still getting a feel for how to do these video conferencing things. So, and to the two people who are here, I just realized that I'm like talking into space. So I apologize for that as well. Okay, so what I usually like to do for this particular meeting is to first sort of talk about some broad principles of a successful grant writer, and then from there move into really walking through how to address the various subsections of standard NIH proposal. Some of you probably most of you are going to be looking at NIH funding at some point. Maybe in the near future, you're going to be looking at small foundation money or other types of grant mechanisms like PCORI. I think so. If you're not going to be submitting to NIH, I think you should still try to pay direct attention to the sorts of things that I'm talking about kind of at a big picture standpoint. Some of the details about the different parts of an NIH grant application may be less relevant to you right away. So the really big picture thing that I want to have you think about really talks about, let's see, I'm just looking at all of the various options of things that I sent here. Okay, this is the first thing that I want to talk about. Really, the real focus of good grantsmanship, it's really as much about marketing as about your science. I just participated in a study that the NIH did that was basically looking at the idea, their hypothesis is always there's an equal amount of high quality science that's going to each NIH study section. While sort of investigators feel there's certain study sections that are harder to get funded through than others. So there was this study that basically gave a whole bunch of experienced grant reviewers grants and asked us to rank them. And they were trying to correlate what study section they were seen in, why our overall ranking. And it turns out that the only correlation they really found is that the highest ranked grants were statistically more likely to be the highest rankings were more likely to be given by reviewers who are most familiar with the science. So it's a human nature type of thing. If you're already convinced that an area is important or interesting, you're going to rank a grant higher than others. And I can tell you that that was definitely the case when I looked at these grants. There were some grants where on lots of measures, I could maybe objectively see why somebody could think that was a high quality grant. And I read it and I couldn't care less about the topic. I didn't think it was particularly important, even though I could objectively see how someone else could think it was important. And that is that's a major thing you need to overcome over and above any kinds of issues with your science. So there's really two issues. There's the people who are going to be into your science, but who score you down because of flaws in your science. Then there's the people that you can submit a grant that is flawless scientifically, but they're just not that into you. And so while you have to write scientifically flawless grants, you also have to address this whole issue of marketing. And this is actually a book that can be a really good introduction to having people think about how to market their science. Mark Kushner is a physicist, actually, who's a PhD level physicist, who's actually also a successful country music songwriter. And he has written many hit songs for the country music industry. And so throughout this book, he sort of uses examples from his marketing of his songwriting to apply that to his grantsmanship. So it's an interesting read if you've never really thought about marketing and what it does for you and how it basically pushes the buttons of human nature to help you get what you want. It's a really excellent read. I don't think it's all that expensive. I don't remember how much it is, but it's not crazy expensive. So that's just something that is worth reading. This is really, this review art or this blog post from Naturally Selective, your grant, the rejected recipe or the funded menu. It's basically going through the same idea. It's using as an analogy, instead of the country music industry, it's using as an analogy of going to a restaurant and deciding about what a restaurant you're going to go to or even what to buy on the restaurant menu. And it talks about basically how you're marketing things versus what it would look like. So I think this is really key. Many grant writers are totally focused on the details of how they're going to do the work, and they don't really look at the key elements of how a grant needs to be, what's going to engage the reader in the not being that into you. Because if you don't convince somebody that your research is important, it doesn't matter how scientifically flawless it is. Under the current grant paradigm, that it doesn't just need to be flawless, it also has to convince the reviewer that it's important. And so this whole thing basically just uses all of these things. But it's he's limited to choosing. So the reality is that if, let's say, you're going into a study section with a 10% funding rate, the average reviewer will have 10 grants to review. We all know this as going in as grant reviewers. And we're going to think about, of the 10 we have, which one or two are the most significant or important. And even if your grant's perfect scientifically, you have to convince the grant reviewer that this is the one that is that really says, this is so cool, this is so exciting. And when it says here is that choose one or two at the most to advocate for. This is really, really important because, especially at NIH, your grant is likely to be given to one person who is clearly a subject matter expert. Then there's a second person who may also be somewhat of a subject matter expert. The third reviewer is likely to be from somewhat out of your area. Like the study section that my grants go to, I am an expert in cataract biology. The study section most of my grants go to is actually a study section that is on basic visual systems. So it is every part of the eye, every part of the visual axis. The first two reviewers are likely to be experts in lens or cataract biology or even eye cell biology. That third reviewer could be somebody who's a neuroscientist studying basic processes in the retina. That my work is not going to be on the top of their radars, the stuff that really is exciting them the most, because it's not in their field. So the two people in my field who read my grant, they really have to advocate for it. They don't just have to like it, say it's good science. They have to strongly advocate for it and convince the last reviewer that this work is really, really important. And so if you don't give your first two reviewers the ammunition they need to say that this work is incredibly important, and you have to be the one giving them that ammunition, that if your first two reviewers give okay scores and the third reviewer gives it, an average score, your grant's not going to get funded. And this is even largely more important, especially at the NIH, is that at the NIH, you have 3 people who really read your grant in detail. They each give scores. Then they present that to the larger people at the grant panel, could be 20 people. Guarantee you that all 20 people on that grant panel are not going to be really into your work. So if the presentation of your work is not convincing or compelling that the reviewers do, that means that there's going to be, people are going to be like, eh. And it's really important if there are people who are giving a range of scores. The people on the panel are going to vote within that range of scores. And so it's, again, not just important to make your science well marketed to your reviewers. It also needs, that marketing gives your reviewers talking points to be able to present that to the larger study section and clearly and concisely say, this is why this grant deserves this score. Because they're convincing people who have never read the grant and who may have no interest in your field of science when that final score is being made. So it's really, really important. And we'll kind of talk about that in detail. Now, this is a file. So the next file I want to talk about that I've given you is, it's called the Do's and Don'ts of NIH Grant Writing. This is probably more don'ts than do's. The history of this file came from, it actually says at the very end of it, there's a professor at Johns Hopkins named Harry Quigley, and he trains medical residents in ophthalmology about grant writing. So he started this as kind of a simple list, and we were sitting next to each other at study section, and he gave it to me. And since then, over the past 10 years, every time I sit in a study section and some applicant does something dumb with their grant, I basically write it down as a don't. So that's why it's more the don'ts than do's. So most of the do's are from Harry. So like have a global hypothesis that solves a big picture problem, use multiple approaches to answer the problem from different sides and so forth. But things like avoid cutesy slang terms which irritate and confuse the reviewer, that's from me. Draw informational diagrams. If you're studying a cell signaling pathway or something very, very complicated, you need to boil down what you're doing in a very easy to follow diagram. These are all hairy because they're very simple, although the wordy ones are mine. That there are certain things about list the total number of publications on your biosketch and so forth. I'm not going to go through this and read it, but I would recommend that you at least look at it. Like here's one that's really dumb, that you always, if you can know who's going to be reviewing your grants, like the members of a grant panel, that people feel sometimes the need to make sure you reference the study section members in your grant application. And the dumb thing about that is that you don't reference the study section's work if it's not relevant, and particularly don't reference it incorrectly. So there's just a lot of little things like that. And I will tell you that there's a real scenario behind every single one of these don'ts. I really saw it happen on a grant application. So the next thing that I want to point out is, this is also a marketing thing. They're just not that into you. This was actually posted on a website that's a blog about ecology, but this is the whole issue about that you often will get back reviews from your grant that you just don't understand, or it seems that what their concerns are trivial, but your score's bad. Sometimes you need when you're a reviewer, you have to justify a poor score. Saying that your work is boring and bleh is not something that one can write as a grant reviewer. So you really, like it says here, I know that if I'm simply not excited by the core ideas of a proposal, I am more likely to get annoyed by stupid details. In contrast, if these ideas really grab me, I'm very forgiving of the details. So it's really important to understand when it's the I'm not that into you versus there's actually scientific criticisms. And this was something that was extremely important for a faculty member in my department. She would always get scores on her NIH grants that they would be scored, but she was never scored high, and the criticisms were rather minor. Well, it turned out when we were able to really unpack what was going on, it's that what she was studying was not that interesting to that review panel. So what she ended up having to do is recast the justification for the project in such a way to pull it away from that study section and have it reviewed by a study section who has a reason to find the work compelling. And that can be hard to do sometimes to get a grant pulled out of a study section, but she had to write the grant differently enough so that it wasn't clearly in this one panel, and she's well funded now. It was really important. So understanding if you have targeted it to the wrong, maybe disease, or to the wrong group of people who are never going to be excited about your work no matter what, that's a really important thing. And that's also part of marketing. And then, let's see, so these are things. This is just I think, quite a well done article talking about being able to write concisely. This is a critical thing because people, especially as grant applications are getting shorter and shorter, and I've been writing grants for 20 years, and every time I turn around, the grants are shorter with shorter page limits. And then they say, oh, we need to make it even shorter. Honestly, the shorter the grant, the harder it is to write. Believe me, grant writing was way easier when we had 25-page NIH grants. Now that they're 13, they're way harder, and there have been people pushing for 6 pages. It would make it extraordinarily hard to write a good grant, particularly when you're trying to convince people who are not that into you in relationship to marketing. What this article really talks about is getting rid of redundancies and wordiness in your grant. I like to talk, and I tend to write very, very wordily. What happens is that you're either going to just write your first draft so you don't put in everything that you need to put in, or they're going to be or you can go the other route, where I would recommend to people, you write the first draft of your grant as long as it needs to be to say everything that needs to be said. And this could be that if you're writing a 13-page proposal, your first draft could be 20 pages long. That's okay. The vast majority of scientific writers I work with, and this is my students, postdocs, other faculty members, and even myself, we all have this tendency to use lots of redundant words. Like the best example of this, it has been shown that in almost every case, you can collapse that to it is. There's really very little need in most cases if there's a reference to say it has been shown that. So, and there's a lot of these sorts of problems. So this article sort of really highlights it. It's basically this paragraph, and it's almost unreadable, jargony thing. And it's a mathematics, it's about teaching math. So it's, and he goes all the way through it, and you can read the article, but in the end, that paragraph that was written by a math education professor is collapsed to every subject presents its own pedagogic problems. So there's a paragraph that's collapsed to one very, very short sentence. And he just kind of walks you through the logic of how that happened in this case. So that's just another thing to be thinking about as you're doing your grant writing. So, and then this one is also really important, is that this is talking about how well do you take criticism. It is, especially for young scientists and even for old scientists like me, it is really important to get other people to read your work. You don't want the first time somebody professionally critiques your work being the grant reviewer. Your chances of getting funded collapse under that circumstance. And this is all about getting over your ego here. And I completely understand not wanting to give this to somebody to read, giving your writing to somebody to read. But you definitely want internal reviews, not external reviews. And this whole article is about that topic. And then now this one is actually the whole issue about how decision-making process is the things that you should never do. Like, this is the top 3 deadly sins in grant writing. You write dense technical prose that is designed only for a specialist to read. Remember what I said, of your 3 grant reviewers for NIH, 1 will not be a specialist in your field. And even specialists are going to be really irritated by too many details and make it hard to read. You don't thoroughly develop your specific aims before launching and writing the text of the proposal. I'm sorry. And this is really, really important, because that one of the biggest problems that I see in grants is that either the experiments proposed do not actually address the big picture problem, or the experiments proposed are different than the specific aims. And there's a lack of coherency between that. And there's a reason that a lot of times what you really want to have somebody do is develop the specific aims page thoroughly, polish it, have a ton of people read it, make sure it's logically consistent, and then make sure that what you actually write in your proposal is consistent with those aims pages. And then this part 3 here, that you basically overwhelm people with technical details, and you don't really make it clear why somebody should care about the answer to those experiments. And that is also really, really a key thing, that you have to convince somebody that you're not proposing the experiments because they can be done, but because they're going to directly answer the burning problem you've already laid out. So, and there's some more details about that. So I'll close this, and then I'll get to, so that was sort of like a overview of some stuff that I gave you for reading. This, though, is the kind of meat of what I'm going to talk about for the next half hour. So I'll pause right here. Does anybody have any questions in relationship to that really quick thing that I just went through? Okay. So if nothing else you remember about this is that marketing is not a dirty word when it comes to science. It's not a dirty word whether you're giving oral presentations, and it's not a dirty word when you're writing grants. So I'm going to go through how to try to work marketing into your grant application, as well as how to talk about this in relationship to the pieces of a standard NIH grant. So the first is the aims page, and something that I find a lot of early career scientists don't really understand is, especially for NIH grants, what is called the abstract in the grant application is probably not going to be read by anybody except congressmen. The abstract is the public document. It's what's posted. If your grant is funded, that's what's posted in the online databases. That's the public description of it. Most of the time, the abstracts are very, very big picture. It's often recommended you don't, in fact, state what your aims are in the abstract. And the reason is that you basically, if you put it in the abstract, then like your competitors can look to see what you're doing. right? It's the thing that is out in the public world. It's a thing that congressional staffers scrutinize, but it's not, very often your grant reviewers will never even read it. So don't make it terrible, but make it talking about the general approach and the general scientific questions you want to answer. Don't say how you're going to do it or the experiments. That doesn't go in the abstract. There's also, this is going to be a very highly cynical reason not to put it in the abstract, is that if you're writing a really big NIH grant, like a 5-year grant, over that 5-year period, you have what you're actually going to be doing experimentally is going to divert maybe significantly from what's funded. And that's just because your hypothesis of one area doesn't play out, while your another aim, you get this exciting finding. So the work is still related to the goals of the grant, but but it's not going to be the exact aims that you originally proposed, because your science is going to evolve over 5 years. That if you put a lot of detail of your aims in the abstract, that's the piece that the reviewers of your competing renewal will see. And then they can directly compare what your original aims were with your progress. And that can look negative, even if you've published a lot of papers on the work. So if your abstract is talking about the general area of research and sort of what big questions you want to answer, and it doesn't go into too much detail, when you're being judged on your productivity, when you're doing your grant renewal, you have a lot more latitude there. Now, with all of that said, the aims page is the key technical abstract of your grant. The aims page is what the study section director is basically going to use to assign the grant to reviewers. It is the piece that the reviewers will read when judging whether to take on the assignment of your grant to make sure they have enough expertise in the area. It's also the piece that will be largely focused on when the program officers are making their funding decisions. These are folks that are unlikely to read your entire application in most cases, but everybody will read the aims pages, all of the important decision makers. Also, the aims page is likely to be the only part that, let's say, 17 out of 20 people voting on your grant will read. So often at a study section meeting, you have the 3 main members who read and reviewed the entire application. When that grant comes up for discussion, the other 17 members will open up your grant, and the only thing they're going to be looking at is the aims page. So you want that to be very, very clear and well communicating. It's, as I said, essentially the technical abstract of the grant, and includes all important hypotheses, justifications, and impact statements. So this would set up the main scientific question, talk about the key data that's supporting a hypothesis, the overall hypothesis, the actual specific aims, and small amount about their justification. And then a very, very important thing about NIH is to make sure you make very, very clear impact statements. So your actual NIH grant score is based on something called an impact factor, or the impact statement. And if you basically aren't obligated to give an impact statement. However, the actual score is driven by impact. So in one of the later meetings, I'll go through an NIH summary statement and explain how the whole thing is put together and how your total score is done. But the only score that actually matters is the impact number. And that number is number 1 to 9. 1 is good, 9 is bad. And that it's not just good science. It's not just whether it's something that should be done. It's that, is the work likely to significantly move this field forward by the end of the grants mechanism? So you can see that for somebody to give a grant a score of a 1 or a 2, They have to be convinced that this is something that is scientifically incredibly important, and you have an approach that is scientifically valid that's going to push that forward. Part of that is you as an investigator. Part of that is your scientific environment. All of these, there's a bunch of sub-scores, but the only score that actually matters is that impact number. So if you can very, very concisely explain what you're doing, why it's going to move the field forward to address these hypotheses, that is really, really critical. Now, the thing that you have to keep in mind is that impact is different than significance, that you will get some separate significance scores. Significance is different than impact because, like, curing pancreatic cancer is significant and important, but that's different than impact. That an impact means that the reviewers are convinced in aggregate, if this work is funded, it will help us understanding the treatment of pancreatic cancer in that particular case. So just because the problem is significant doesn't mean that you're going to get a good impact score.
And is part of that they feel like the project may be significant, but in the context of the investigator, the environment, productivity, feasibility, those are the things that then contribute to impact?
So the scientific question can be significant, but it could be that all of those things are fine, but the part of the problem that the investigator has chosen to investigate is unlikely to play a significant role in solving the big picture problem. So everything you just mentioned is absolutely on the table for a poor impact score with high significance. But it also can be that the experimental plan will not really, the information we're going to get out of that is unlikely to move the needle. So it can be any of these things. But this is, again, the marketing side about crafting your impact statement, is that you want to give your reviewers ammunition to easily lift out of your grant. If they think it's wonderful, give them the talking points they need to sell it to the rest of the study section. And even if somebody reads your grant and loves it and it's their area, this is somebody who's doing this on top of their regular job. They're often doing these reviews, after the kids are in bed at 10:30 or midnight at night. They have just flown in from the West Coast. They're tired. They have 7 grants to discuss orally. And the easier you make it for that person to advocate for you, the better off you're going to be. So articulating impact statements is important. Now, so that's the first page. And then the other 12 pages of a standard R01 grant have these 3 standard sections, and that is the research plan. Now, the first is the overall significance. So this is your intro to the field, and that here you don't want to necessarily talk about the significance of your individual aims. That you save that to talk about when you talk about your approach. The overall significance is putting together the big picture of the area. Now, in the current world with the current grants, this is not a lit review. Think about this as like if you were giving a talk on your work. You don't give a comprehensive literature review. You basically say, this is my general areas. Like, let's say I'm studying speech pathology. Speech pathology is really important because kids who have speech pathologies have these psychosocial issues, blah, 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 blah. That we have these really big picture questions about what we're trying to fix. And some of the competing theories, we have evidence suggesting this theory is the most important. And that we're going to address this in the specific aims of the grant or something like that. It stays very big picture. Now, some people choose to put the more detailed significance here, but it's more effective to put it in the approach section under your aims. And the reason is that it's a human nature thing. Even a 12-page single-space proposal, the people might not be reading it in one sitting. Grant reviewers are likely to be highly distracted when they're reading your grant. Maybe they're sitting in their office, they have students coming in, whatever. So you need to design it in such a way that somebody with a reasonably short attention span can get through the whole thing. And I'm saying this as a grant reviewer. I'm not picking on grant reviewers. This is just the reality of the people they have doing this. So it's this, the reviewers are unlikely to read your grant in one sitting and to expect them to remember aim-specific information. One thing that early grant writers often find very frustrating is in the section of their grant about their aims, the reviewer will say, well, the PI didn't address XYZ when it was addressed earlier in the application, but you can't expect a reviewer to be hopping around the application. They need the information when they need it.
So when you say in the aims, you mean in the approach section?
The approach
when
section.
You talk about
the approach section. So the overall significance section in a 12-page grant is often a page. It's a pretty short section. Actually, sections A and B together may only be a page. It might be a little bit bigger than that, but it's still not very long.
But you'd still talk about the fact that there are gaps in what is known.
True.
Right.
But you don't dive down real deep into justifying their
like a really specific thing.
Because those gaps in that really specific stuff, you do talk about it. You just put it in the approach section. Because if you don't want the approach section to just be a list of experiments, the approach section should give the scientific justification for each experiment, what it's going to tell us and how that's going to drive the field. And that could be your preliminary data supporting why you're going to do that experiment. That should go in the approach section, because you don't want to put all of your preliminary data, let's say, in the overall significance section, and then make the reviewer hopping around. I guess what I'm trying to say is that structure a grant like a good talk, less like a scientific paper, where like in a scientific paper, all the results are crammed together, and in a lot of journals, you're really discouraged from talking about what they mean, and then discussions here. But if you gave a talk that way, it would put everybody to sleep. Writing a good grant is probably more structured like the talk, not like the scientific paper. Does that kind of make sense? Okay. All right. So then what I basically when I explain this concept to my grad students doing talks, I say, don't front load too much information, because you're going to lose your reader in this case if you do. All right, innovation. Now, to be honest, personally, I'm always sort of frustrated and irritated about this section. But to be perfectly honest, for many reviewers, this is a major driver of that impact score. And you're basically trying to say, why are you different than the other 10 people interested in this area or going after this problem? So what is different about what's proposed here compared to what others are doing in the field? Now, it could be that you have this great new technique that no one else in the field has ever applied to this problem. So you're saying you're different because you can answer this question using this great method that's never been done. That's fine. You can also, but it also could be an innovative idea, an innovative hypothesis. it could be that you've developed a new technique that can answer something that has never been answered before. But it's important to clearly state what is innovative. For some reviewers, innovation is a major score driver for that final impact score. And you don't want your reviewers to mark it down for being boring. Now, this is, again, a marketing thing, because what is innovative to one set of reviewers will not be to another. I'll use, this is a diverse group, but something that's kind of hot in my field is CRISPR-Cas9. It's the new gene editing technology that's used to do gene editing. A lot of people who are like working from human stem cells, being able to gene edit in human stem cells and do studies. It's a really innovative thing. But if you're in certain fields, they've been using CRISPR-Cas9 for 5 years, and it's not going to be innovative there. But if you're in another field where, let's say, you're interested in, the corneal endothelium, and you're interested in certain kinds of corneal diseases, I don't think anybody's ever published using these methods to answer certain questions. You might get a lot of innovation scores there because you're bringing in this technique from other fields to answer this problem for the first time. So innovation, unfortunately, is a moving target, because when it's innovative, something that's innovative today might not be innovative in 3 years because it's become the standard in the field. It is important to really think about, though, is that why are you different? Why should you be funded and not, the other people who are working in your area that, might have presented at the last conference? So what sets you apart in either your ideas or your technology? So that's an important part of the application. All right, so then we'll go talk about approach. So an approach is that the way they describe it, it sounds like a description of what you will do to achieve your aims. However, the approach section, even from this description of what you're going to do, you have to address ideas on data analysis and interpretation. It's really important to acknowledge problems and experimental limitations. If you say, I'm going to do it this way for this reason, that there are these limitations to what this can tell us, or these are reasons that this will fail. You want to be the one to point that out to the study section. You don't want the study section member to say, well, that they're being really naive and they're saying that they can do it this way, but it can fail for these 4 reasons. If you bring it out, you look savvy, because we all know that there's no perfect experiment. If you don't bring it out and the reviewer does, that means you're naive. So it's really important to always have problems and pitfalls, alternate strategies, and benchmarks for success. So benchmarks for success are if your hypothesis is correct, what is the expected result from an experiment? And then you say, well, if we don't get this result, it would mean X, Y, Z. Don't write your grant like you're assuming your hypothesis is right. That really sets some reviewers off. Don't just say, I'm going to do this experiment without describing what the data would look like that supports my hypothesis and what would be the meaning if I'm getting the opposite data. So that's a really important part of this. And you need to be objective and very critical. Always remember that correlation is not causation. Like in my field, people will say, well, if a gene is expressed in a certain spot, that tells me it does X. But that's not true. You can get rid of certain genes and everything's fine. So correlation is not causation. Don't overinterpret what your experiments tell you. Also, make sure that your experiments actually address your big picture problem. Often people can write these very, very elegant descriptions of their big picture problem, but in the end, that the experiments being done don't really address that big picture problem. They might address something else. There's this disconnect there between the problem stated and the experiments and what they're going to give you. So that's a really important thing to, after you've written your approach section, really objectively say, is this answer going to really address the problem that I marketed as saying is my important problem? Now, some things about approach that I sort of alluded to up in the significance section, that don't just make this a list of experiments, that you need to justify saying that that this is our problem. Our preliminary data says the answer is X, but we haven't proven it. So we need to do experiment Y, which will tell us whether it is really working this way or not. This is where you would put that preliminary data. I'll take an example from my work, is that you see a gene expressed at very high levels in a tissue. You're assuming that that gene's going to be functioning there. And we want to really show that by removing the gene from the tissue. So if we're going to set up that experiment, we would say that, we're really interested in this biological problem, that we have preliminary data showing that this gene that does this function in another tissue is expressed at really high levels in the location I'm interested in. That's where you show the figure with the expression study. And so that they have the preliminary data that correlates with your assertion right next to it. You then can say that the next step is to determine whether this gene is actually functional. We need to do these approaches to delete it.
I have always a question about the preliminary data. Is it okay to put the unpublished?
Absolutely. Just keep in mind that it sort of depends on the grant mechanism. So for an R1, that published or unpublished preliminary data can be put in. Now, from the standpoint of published preliminary data, your own data from your own papers is considered way stronger preliminary data than from somebody else's work or somebody else's investigation. For unpublished data, you have to keep in mind that make sure it is your best approach. The advantage of published data has been through peer review. Unpublished data, the problem is that it hasn't been through peer review, and very often people are kind of sloppy with it. So they'll show things that are very poor quality or have not been, in my case, like I do a lot of histomorphology. So that I can't show a non-publication quality image in the grant. It has to be highly convincing and highly high quality. If you show an ugly image, then the reviewer is going to say, well, they can't do this at high quality. So I lose confidence in what's going on. So the quality of the data in the grant has to be suitable for publication, because showing poor quality data in a grant application is going to reduce the confidence that the reviewers have in the strength of the preliminary data supporting the investigation, as well as, especially for a young investigator, their confidence that that the investigator is capable of producing such high quality data. I would assume, like in your case, it might be really important that statistics are really, really solid or done correctly. In my case, it's making sure that, what we're showing is unequivocal. The morphology of what we're showing is high quality. But we have to do that convincing. There's actually an experiment in the paper that I'm writing right now that we did it entirely for the grant initially, because we had submitted the first grant, and the way we had presented the data, the grant reviewers went after us and they said, oh, this isn't convincing at all. You need to put both markers on the same picture. And my grad student actually spent 4 months getting 2 separate probes working simultaneously on the same piece of tissue. And we wouldn't have put that effort into it, but the grant reviewers trashed our grant because we hadn't done it. She finally got it to work. That picture went into the grant, and that picture is in the third figure of the paper we're writing now that we had gotten the grant funded and we finished the project. I mean, it was publication quality, and if it had been poor quality, we wouldn't have gotten the grant.
Can you talk about where preliminary data would go? So I'm assuming from what you just said, it does not go in the significant section.
Usually
because that's
not.
Much broader.
Yeah, usually the best written grants. I mean, this is all advice, right? It's not in the rules. My advice would be, it depends. So if you have like a really big picture piece of preliminary data, that can go in the significant section.
Okay.
However, if it's the preliminary data that justifies that the next experiment needs to be done, it goes in the approach section. So like if, I'll, for instance, of a grant that I'm going to be writing soon, that we have found a gene expressed at high levels in the lens. We've gotten rid of it from the animal, and the animal has an incredibly fascinating lens phenotype. Okay. Now we have to figure out why that's happening molecularly. So in the first aim, we'll have the expression pattern. That'll be part of the figure. We'll have a picture showing the phenotype. Then we're going to say that the next experiment needs to be done is to fill in, the natural history of that phenotype, the whole etiology. And so that these pictures will be just the preliminary data justifying why we're going to do the detailed histomorphological analyses. Now we already have a couple genes whose expression is off. In aim 2, where we're going to be talking about molecular mechanisms, we're going to basically say, prior studies have suggested this gene is doing these functions in the immune system. We've already found.
Becky, I saw your email to Cynthia. So I guess I was confused because I think we thought we were doing the coffee hour this coming weekend, but somebody is doing it this Sunday and it's not us?
I think somebody's on the wrong conference call. This is a grants writing workshop? Okay. Don't know what's going on with that.
They muted. I think they were on our call. But they couldn't.
Yeah. So essentially, the next aim will be saying that in the immune system, this gene does these things. We've shown that there's this molecular change that is consistent with the function in the immune system. Now we want to see, is this function really, really important for the biology of our tissue? That data, that expression data will be buried in that aim.
So, and I obviously was thinking about the approach in the wrong way, and then I was thinking that it literally was describing the methods and that you wouldn't have all of this in there. So can you just give us an example of like how it would be structured? Like you'd have aim 1, and then maybe you'd have a paragraph.
A paragraph saying, right, exactly. Okay. So you have your significant section is the really big picture. Like
right.
In my case, it would be, talking about, we don't really understand a lot about age-related cataract, blah, 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 blah.
Right.
We know these genes. We don't know a lot about the genetic basis. There's this approach for doing it. We've pulled this particular gene out for further study. And this whole entire grant is based on focusing on what this gene could be doing. Okay. Then when I get to the aims of this grant are basically going to be really figuring out the histomorphological characterization and the natural history, the molecular mechanism of how the gene might be causing this natural history. And then I don't have my aims developed yet, but maybe the third aim might be something. I might only do 2 aims for this grant, but the third aim might be something like, how does this gene interact with other genes known to be involved in this process? Something like that. I don't know. So that's where I would then dive deep, justifying why I'm doing these type of analyses. I might show preliminary data from another project showing that we know how to do this method. And so the type of data we would get would look like this. We're very competent at that. We published 5 papers on it. Here's an example of what this data would look like. That's another kind of preliminary data that's not on this project. It's simply showing that in your hands, you can do the experiment proposed. Because the problem is if you jam all your preliminary data at the beginning,
Right.
you're expecting
People to remember.
And well, not just that, you expect them to be able to relate. It's important to also explicitly explain why this experiment will answer this question.
So you would say, okay, A1, and then you'd have and you put the hypotheses right there?
Oh, well, every single aim should have a hypothesis,
Right.
a sub-hypothesis that fits into your global one.
Right.
So, and it even can be, you might even have a separate hypothesis for every experiment you're proposing. Because very often the reason you're doing the experiment is usually, you're hypothesizing a particular thing. I mean, there are some experiments in my field where it's called hypothesis generation research. Generally, those don't do well with study section. Some classic things are like global expression profiling and stuff like that. Generally, you want to avoid proposing hypothesis generation research in a grant. So almost for every experiment you're having, because if you don't have a hypothesis, then you can't explain what the expected outcome of the experiment is. And then you can't directly address what you will do if your hypothesis is wrong. So, using the H word a lot in a grant isn't a bad thing. That sort of makes sense.
Mm-hmm. I have a question about the number of specific claims, because it's usually people put the 2 or 3. Is there anyone who actually done a 1 or like?
Well, it depends on how long your grant is. I mean, that most of the grant writing I've done is R01s. Now, usually if your grant is 1 aim, you're going to be having sub-aims. So it's easy enough if you say, I have 1 aim. Well, if you have 3 sub-aims, just break those into 3 aims. It might be that the way you've drilled down is not really right. I have written and had funded grants with 2 aims. Sort of the classic NIH grant is 3. But you're better off having 2 aims than having the 3rd aim be weak. Because something that happens a lot is somebody will write 2 really good aims. They run out of steam, but feel they have to write a 3rd aim. And by including the weak 3rd aim, they lose the grant. Because the study section is not, and very often the study section says, I really wish I could just say, dump aim 3 and, suggest this be paid for aims 1 and 2. But study section's not allowed to do that. The score is based on the entire grant. So from that perspective is that if you don't have a strong 3rd aim, don't include it. Grants with 5 aims also don't do well. That a lot of times is because it seems that the project's being, drawn out too much. I mean, often I'll write grants with 2 to 3 aims, and each aim will have sub-aims because they're the separate experiments. You do have to be careful that your aims are sort of independent from each other, so that if 1 aim fails, your next aim isn't dead. So that often is a bad thing for grants because, you're not going to give you $1 million and at the end of year 1, you have nothing else to do because aim 1 failed and what you're going to do in aims 2 and 3 were dependent on aim 1. So that's sort of like in my case, my histomorphology is independent to my molecular work. So it's more, think about it from a human nature perspective. I mean, I think you have kids, right? Mm-hmm. Yeah. I mean, just imagine a hard day in the clinic, you're dealing with your kids in a tantrum, and then now it's like, oh, my grant readers are doing 3 darn days, and I have 7 to do. I'm going to be up to 3 o'clock in the morning, and then you have to be on rounds 5, right? And you do that for a couple days in a row. Just think about what your attention span's like. So, and it's similar like if you go to a seminar and somebody, says, big picture question. And then they show, 20 slides of experiment. And they're expecting you to understand why they did the experiment based on something they said about the introduction 25 minutes ago. And then then they give a discussion of what the data meant. Even if you were in the field, you're not going to be following this. You want somebody to sit there and tell you a compelling story, and don't front end too much information.
This is extremely helpful. I have never actually heard this before, and I just think it's so helpful. So thank you.
Yeah, I mean, it's something that the best grants do this.
Wonderful. Yeah. Very helpful.
So let me think something. Okay, so, and then the last thing I will want to emphasize is that make sure that your compelling arguments in the significance section, saying that we have these holes and we need to answer them, make sure your experiments really are answering that problem. And it's really easy because you tend to write on grants on sections, so your ideas and your thinking evolve. And also back check to make sure your final approach section is coherent with your aims page. Because very often you'll put a lot of effort into your aims page, and then you're writing your grant, and as you're reading more literature, as you're really thinking about it, your ideas evolve, and then the aims page isn't updated. And those sorts of disconnects really annoy people.
Do you put your hypotheses in the aims page?
Yes. And it's actually not a bad idea to state your aim as to test the hypothesis that such and such is such and such.
Okay.
Generally, there's different styles about how aims pages are done. Personally, I would recommend not putting your experimental design on your aims page. That I usually recommend to say, like, aim 1 to test the hypothesis that such and such is such and such. In the little commentary under that, something like, we have previously shown that X, Y, Z are true, which implies that Q is true. That you can, very briefly say, like, so we're going to investigate the structure of knockout mice for Z to see if Z controls Q, something like that. But it's not an experiment vomit, the aims page. That usually you want to avoid wishy-washy things like to investigate or to determine, but sometimes if you don't really have a strong hypothesis statement to be making. But you could say to characterize such and such and such and such, and then in that little description say, we hypothesize that this affects that, so we're going to. But, don't, and also you can even like bold the hypothesis word or italicize it. You want somebody to be able to look at your page and very quickly pick out hypothesis and, objective. I mean, these are things that a quick scan of the eye. One of the faculty members that I mentored, most people don't do it enough, this kind of highlighting. You can overdo it. One of my mentees who, to be honest, he is a superb writer. He was a superb writer the day I met him. He's really, really good at marketing his stuff, and he gets a lot of recognition because he's really good at marketing. But he overdoes boldface, because if you have too much boldface, it no longer sets anything apart. So usually when I read his stuff, it's like, all right, kill the boldface already. But most people don't overdo it. If anything, they underdo it. Now, this is something new, which I'm still trying to figure out exactly what they mean here. They just changed the NIH grant rules. I think that this is going to be important might be for this February, definitely by the June submission, this is going to be critical. And that is reagent integrity issues. So there has been a bunch of reports about, and it's basically drug companies complaining that they're unable to reproduce results coming out of academic labs. There's also, issues with reproducibility between academic labs, but the really strong things have been drug companies trying to reproduce certain key findings in the diseases they're interested in. And often these are going back to problems with integrity of reagents. And this can include the actual genetic background of animals, cell lines. There's a big problem where, I don't know if you're familiar with, there's a famous cancer cell line called HeLa. There's a lot of bioethics case studies about it. It came from a cervical cancer patient in the early 60s. HeLa cells are so aggressive and malignant, they actually can cross-contaminate cultures via aerosols going through the air. And they grow so fast that if they do cross-contaminate, they can take over cultures. And people were thinking they were studying pancreatic cancer or certain other things, and they were studying HeLa cells. So now there's new guidelines. A lot of journals are requiring it. Now NIH is requiring it, that you have to validate your cell lines genetically every certain number of generations. And you have to describe for certain journals how that was done. Well, NIH is starting to require that now. So you have to address the integrity of reagents, of your cell lines, your animals, your antibodies. That's a really critical issue because a lot of antibodies that are used in research, they have kind of iffy specificities. Also, reproducibility of experiments. And this is the good statistical plans. Especially those of you who do social science research, this is going to be really, really scrutinized because there have been so many cases of, I think a famous one was, I think they went out and they were looking at whether or not, this one might have been actual research integrity, but there was a famous one about if you were interviewing, if you send somebody out to directly talk to somebody about gay marriage, they're more likely to accept it. And it turns out the results completely irreproducible. Now, I think that one might have been a research integrity thing. The guy faked the data. But there are plenty of social science things where they weren't actually correctly analyzed statistically, or they weren't powered correctly statistically, so that you got one result because of random chance, not because it's a real way people are. And so for social science research, that reviewers are being told to really scrutinize the statistics involved. I'm an editor of a journal that there was a paper that just recently got bounced because it was in statistical genetics. And it turned out that the study was so way underpowered. They looked at 1,000 people, and the statistical geneticists said to make the findings that they had actually real and not random chance, they would have had to look at 500,000 people because of the way complex genetics work. So this is going to be very highly scrutinized in the future if that's your kind of work. Also, this is something that you don't really state in your grant, but you should be able to and it's good if you can get your application written enough in advance that you can put it in the drawer for a week, then step back and look at it again. And objectively say, if I did all of these experiments, is this going to be publishable? Have I proposed what would be needed to publish the work? At least one paper, hopefully more than that. Because if you answer the questions, but there's still going to be huge holes that are not publishable, these are going to be the same holes the reviewers are going to be looking at. So it's another way to kind of gauge. And also, to be honest, the only outcome that matters is a publication under most circumstances, unless you're doing some kind of like population-based social science or intervention where you might be changing public policy. In most cases, it's a publication. And even in those cases, it would be a publication to say, we did this intervention, we fixed the population in this way. You want to publish that and disseminate it. So objectively step back. Now, this is also that anything in early stages of development, it's really important and like this is really important for pilot projects, your strategies to establish feasibility, also managing high risk of the work. For instance, if you've never done the experiment, make really sure you have a appropriate experimental support. Well-known collaborators can be really, really important here. One of the things about the well-known collaborators, they have to be convincing collaborators. So that ideally, the convincing collaborators in the door next door, and you've already published with them. If that is not the circumstance, you're going to have to really, really sell that it's a convincing collaborator. And convincing collaborators are seldom from outside the institution. I just recently published a paper with a guy in Australia, and we work very well together. Now I can probably sell in a grant application that he's my collaborator on this because we've now published a couple of papers together. But if I just proposed him as my collaborator from Australia, it would look, even if I had a letter from him, there's a lot of fantasy collaborations out there. So if the collaboration is important, you have to be convincing the reviewer that it's really going to happen.
Well, for K awards, for like a mentor, you're trying to find the person who is sort of the most prominent in the field, but they may not be at your institution.
That's another case where it's really important to be really convincing. Okay. Some things that are crucial here is make sure that that person has really read your K award application. I've sat on plenty of K award panels where that they had this fantastic on paper mentoring team, and then the application had flaws, and everybody's like, "There's no way that these mentors read this application, because if they had and really took their mentoring responsibility seriously, it wouldn't have had the flaws that it had upon submission."
So it suggests that they're not as engaged as.
Because there's lots of people who will write a letter, but that's very, very different. There's another K award that I can think of where that it was that they basically picked a lot of prominent people in the global field, but they weren't appropriate mentors for the project. So it's like, well, and in this case, this is from my field, and I was like, and the one guy's name is Joe, is this really prominent guy, and the study section was sitting there, it's like, why in the world is Joe her mentor? He's, he works 100 miles away. His scientific expertise is not related to hers or her project. And it was just, she basically put him on as a really high profile person.
So is it better to have somebody who is like extremely high profile, but that maybe you haven't published with before, or you haven't worked directly with before and shown that relationship, and it's for a K award? Or is it better to have someone who is, accomplished, high profile, maybe not the most, but that you've published with?
Okay, so it's more mentoring teams, right?
Right.
And that each person on the mentoring team should be identified explicitly on what strengths they're bringing to your mentoring team.
Right.
So the thing about high profile is you have to be careful about is that they're also really busy.
Right.
So it doesn't matter if you have a Nobel laureate on your committee if he never talks to you, right?
Right.
So it's a happy medium. Like some of the best of these mentoring team plans are like somebody is interested in a particular scientific problem, and they're using advanced genomic techniques to answer it. So they have their postdoc mentor as their K award person. Then they have a prominent bioinformatics person to help them do the data mining in their genetics work. Then they have somebody who's really good at techniques development. Let's say if the idea is that if they pull something interesting out, they want to set up a genotyping platform for that disease or so. So an expert in development of genotyping platforms, an expert in bioinformatics, and an expert in the disease, let's say. That's a mentoring team that basically each mentor will fill different gaps in the training of the applicant.
And that makes a lot of sense. I guess what I'm wondering is when you're picking that mentor of that particular area on that team, is it better to pick someone who is well published, well funded, and you've worked with before, or is it better to maybe pick someone who you haven't worked with before but is maybe the most prominent person in the area?
I would say first one you said.
Okay.
But you have to be careful because if you've already learned what you need to learn from that person, you have to be saying that what new information am I going to get out of that mentor I've had the 5-year experience with, right? It could be, I mean, you still talk to that person and they'll still mentor you, but they're not filling in a hole. You know, a lot of the K awards, I haven't reviewed K awards for I always forget the numbers, but most of the K awards that I've reviewed are either grad student postdoc or the transitional K to R award things. And in that case, the primary mentor is the person's postdoc advisor. Right. And then the secondary mentors are people who have expertise in the work being proposed in the research plan that is independent and separate from the research mentor. Mm-hmm. And when it's strong, it's maybe people who already have a working relationship with that lab, or folks who there's this demonstration that there's already some mentoring relationship involved, but that it needs to be more sustained. In some cases, if the person's in another institution, then it's can be helpful to say that, oh, I'm going to go and spend 3 months working with so-and-so to learn this technique. You know, but you have to be careful about it being convincing that it's really going to happen and isn't going to see.
That information of the collaborations or mentors or contributors, additional members. How much reviewers are going to pay attention on the things written in the biosketch? Because there's a NIH style change last year.
It's very important. This is also part of the commitment of your mentors. Okay. So the problem is that, and this has actually been really, really obvious, that if there's a committed mentor, they're going to reformat their damn biosketch. If there's not a committed mentor and they're just writing a courtesy letter or signing a letter that you wrote, then they're like, oh, I'm busy. Here's my old biosketch. It's not going to fly. It's going to hurt your application because it's not going to be a convincing mentoring relationship. And also in the new biosketches, I think I have this right because I think I have. Yeah, I did redo my biosketch for last summer. That the top of that is basically like I think it was actually a mentoring one that I basically wrote that to say why I was qualified in my current relationship with the mentee. So if the person can't be bothered to explain in their biosketch why they are an important and appropriate mentor for this individual, that's almost saying that that's not a real mentoring relationship. And if you're asking somebody to mentor you and they can't be bothered to write a paragraph explaining why they should be mentoring you, you're not going to have enough input from them to be a viable mentoring relationship. That's how I would look at it as a reviewer. And because that's the sorts of things they look at in these applications. You know, is this going to be a real mentoring relationship? And it's just, you know, it could be that person, you know, had a death in the family and 10 billion things happen right then and they couldn't redo the biosketch. But it's considered evidence that it might not be somebody who's engaged.
So it's better to put the redundant information in a biosketch as well as in, let's say, like approach section.
Well, I mean,
there's a lot that's going to overlap.
Well, sometimes. I mean, some of it is a chance to elaborate, right? I mean, like in the biosketch section, you're basically explaining why you have the expertise to do these things. And you might show a piece of preliminary data in the approach section saying, I've done this experiment before. But to say that, you know, I am well qualified to do this work because I have done these experiments and I've done X, Y, Z, P, Q, you know, that is a kind of an important thing.
And they are reviewing the biosketch. I know you said everyone's busy and they're tired, but like the biosketch is something they're looking closely at.
Yeah. Okay. I mean, I don't actually know how the brand new biosketches are being looked at because, in my opinion, as a person who's been reviewing grants for almost 20 years, I find the current biosketches way less useful than the old ones. And if you're too verbose, is a reviewer going to read those 3 paragraphs? Probably not. But, I actually personally, this is my personal opinion, I don't like the new biosketch format, but I have no power to change that.
So much.
No problem.
Incredibly helpful.
Is there any questions from the people out there? We were just kind of having a conversation in the room, but I'd be happy to entertain any questions from people who are out in the universe. Because if not, we're probably going to shut down. Okay. Actually, just one thing I do want to say is do not end the grant application on a pitfall. The very last thing they should read is basically, again, a sales paragraph saying that if I get all this stuff done, I'm going to move the field in these ways. And, you wouldn't say it this way, but that's why you should give me all this money or give me a great grant score. You don't want the very last thing the person reads in the grant to be a negative. And, so that's kind of an important thing as well. So with that, I think we'll shut down.
Melinda, we actually have a question by chat.
Okay.
Someone was wondering about distinguishing the aims first page and significance of second page.
Okay, so you're talking the specific aims page should be completely readable, independent of the rest of the grant application. So really think of that specific aims page as a technical abstract. It should be readable and interpretable and followable without reading the rest of the application. Think about the significance, the innovation, and the approach section as the body of the grant. And actually, if you even look at the NIH grant guidelines, they don't consider the specific aims page as part of the main body of the grant. They're actually mentioned separately among the page limits. So that in the significance section of Section A of the research plan, you're going to be elaborating. The specific aims page, you're going to make the same big points, but you don't have the space to elaborate like you do in the main body of the grant.
So there will be redundancy between the two?
Absolutely. Because the specific aims page is really an abstract. It's just like if you were writing a paper. The abstract of your paper is fully redundant with the rest of the application. If that makes sense. You should consider it in the same way.
Great. Looks like you answered his question. Does anyone else have any questions? And if your microphone's not working or forget how to turn mute off, you can also use the chat option. Okay, if there's no other questions, Melinda, thank you for your time and thanks for staying with us longer.
Okay, and hopefully I'll get to meet people next week at the Excel conference, or I guess it's the IDEA conference or whatever the conference is next week. Yes,
correct. So if anybody wants to meet Melinda, you'll see on the agenda there is an MRDA meet and greet. But if you don't have an MRDA, please, that does not exclude you at all. Just grab your box lunch, come on down, come meet Melinda.
Okay, and I'd be happy to also, if you have a chance to look at these things, I'd be happy to, sort of elaborate on some of the things that I've seen over the years and, if nothing else, tell you some funny stories. Okay, so hopefully I get to meet a lot of people next week.
Melinda, thank you, everyone.
Okay, bye.